the technique of genomic selection as a routine practice and currently working on developing strategies to purge genetic load from cassava clones. May I please welcome Dr. Ramu Punna, Genomic Selection Application Coordinator and Research Scientist at Corteva Agri-Sciences for his demonstration.
Good afternoon. So today I'm going to talk on genomics selection in plants. So actually I would like to give the demo rather than giving a lot of talks here. So everybody is talking the genomic selection and its importance. Already Babu highlighted, and yesterday the Philomin showed the case study in wheat, how this is helpful. So most of the slides, they are repetitive, so I try to skip a lot of slides, and where it is needed I will explain, then we'll go to demo. So in any breeding program, normally any breeding program aims at creating the variation to develop the new plant varieties. So this creation can be done different ways. Either can be recombination, or it can be creating the random mutagenesis, or yesterday Raman Babu explained about the disruptive breeding technology like CRISPR-Cas. So whatever the technology uses by breeder, ultimately they end up with developing a population. So once they develop the population here, they need to select the superior lines. Morning, Babu asked, how do you select this top 5% of individuals in your experiment? So that process is called selection. In this exercise, we are mostly taking the help from the genotyping data. That's why it is named as a genomic selection. It's not only the genomic data, it also uses the phenotypes, but mostly it's guided by the genomic part. So here the breeder's equation and the genetic gain already explained yesterday, so I'm just skipping this one. And so after the invention of the molecular markers, the plant breeding community has benefited a lot. These impacted and accelerated the plant breeding, and they accelerated the genetic gain in every plant breeding. So here this a plot I'm showing here is a 4-dimensional plot. So x-axis is the effect of the QTL, the left is a low QTL, and the right side is a high effective QTL, low effective QTL, and left is high effective QTL. And here you have the elite germplasm and unique germplasm. If you are identifying the markers to selection, or if you are identifying the QTLs, mostly you are end up with here. You end up with few number of loci, and you are having the very few individuals of the interest. So as you move from left to right, as your QTL effect is very less, then you don't have markers supporting for that particular QTL. So you need to use the more number of markers, and you need to use the more number of individuals. So the genomic selection getting the importance in this context. So here I would like to give what is the difference between the marker selection and the genomic selection. As you already know what is the marker selection. So here this is a process. You develop, you identify the parents and develop the mapping population and genotype and phenotype and do the QTL analysis. So in this QTL analysis, you end up with either identifying the 1 or 2 large effective QTLs. If the trait is simple, you get 1 or 2 large effective QTLs. If the 1 or 2 large effective QTLs, they are flanked by the markers, so you can use the marker selection program very effectively if that is the case. But in reality, so if you're talking about the plant yield is not controlled by 1 or 2 QTLs. It is controlled by the large number of QTLs, but the effect of each QTL is very, very small. So you don't have a significant markers associated with the very, very tiny QTLs. So you need to use the large number of markers. That's why we are getting the genomic selection. So the marker selection is very effective when the large effective QTLs are identified. If not, only the option we could see is the genomic selection. So in case of marker selection, even if you have the 2-3 very good QTLs, it takes a lot of time to pyramiding all these QTLs into one program. So to avoid all these things, the genomic selection is getting the importance. Selection, you don't need to know where these markers are lying, whether this is associated or not. You don't know. You don't need to know. So you can go ahead and use the marker data for the genomic selection. But in case of marker selection, you have to know where this QTL, you need to know where the genomic, you need to know linkage map, you need to know QTL map, and what are the markers linked. This is all prior information is needed. But in case of genomic selection, you don't need to know anything. You just simply do the estimate, you try to estimate the SNP effect and just proceed. And here I'm showing the standard difference between the normal breeding program and the genomic selection program. As genomic selection program getting the impedance to reduce the cycle. When they reduce the cycle, that means in a breeder's equation, the denominator is the number of cycles in a given in a year. So if you reduce the time per cycle, so it will increasing the denominator. You are decreasing the denominator. That means you are ultimately increasing the genetic gain in your breeding program. So in the normal breeding program, to release a variety, it takes 10 to 12 years. But in case of genomic selection, you are able to release the varieties between 6 to 7. So you are saving almost like 5 to 6 years in the genomic selection. So here in the genomic selection, you need to develop the training population and you have the test population. So you develop the training model, then you pump all the testing material into the training model and estimate the effects and the GB values and select the individuals and move on. So why this is getting importance? Because phenotyping is very expensive and uncertain. It depends on the environment, and genotyping becomes more affordable and cheap and become high throughput. And nowadays we already have a lot of the sophisticated statistical models to do the genomic selection. And the computational power is increased as compared to maybe last 30 years now. If you go back and see in the last 50 to 30 years, we have very small, powerful, small computers like 1 gig RAM, maybe 512 MB RAMs. But now there is no limit to have a RAM. You can have how much you want you have, you can do this. So here I'm showing a lot of the genomic selection models are already developed. I'm just giving a gist of examples here. I will just show you the Bayesian model, how that can be run using example data. So if you want to see how each model is different in its own way, so each model algorithm is different. So when you do when you run the same data set in a different models, it end up with a different genomic prediction accuracies, because how they shrink the model or how the algorithm works is different for each and every model. So here I'm showing an example. So this is RR BLUP SNP effects here, and the Y column is the base CPI model SNP effects. You see the SNP effects varies from -1 to 1 here, but in case of here, say -1 to somewhere, say it may be -5 to 12 or 13. So the scale is different, but in both cases, they are able to capture the large effective QTLs and they are able to capture the small effective QTLs. So so it doesn't matter which model you use this, but it matters sometimes. It depends on your material and it depends on your crop. So it has to be taken care when you are running the genomic selection model. And this prediction accuracies depends on many factors. One is what is your population size. Another is the trait heritability. Trait heritability, if you see yesterday's Philomene example, she has a very high genomic selection accuracy, because that is controlled by one or two genes. It's a simple trait. We need to understand how the data format is. Yeah, we'll next. First BLUPs. Yeah. So first we need to understand what's the data format look like. So for example, so this is the phenotyping data format I'm going to show you. So I'm going to show you the two traits. One is the color, another one is the grain yield. So color is simple trait. I coded either 0 or 1, whether they have a color in the grain or not having the grain. And here in case of yield, we have the high-5 BLUPs values. Here already we adjusted the phenotypic values across the locations to calculate the BLUPs. Okay, so here I'm already showing here is already adjusted values. So I will give you a demo how it can be done running the BLUPs. Same thing. So for calculating the BLUPs, so I'm going to use a software called MetaR. This is developed at CIMET, and this is a publicly available and freely available. So anybody can download to your system, and I gave the link as well, so you can just simply download and do this. Yeah. So I'm not going to give why we are doing the BLUPs, because already in the morning Babu explained why we are going to estimate the BLUPs.
Okay, just wanted to give you some context to what we are doing here, BLUPs. So we discussed in the morning, no? So if you are doing, let us say, a trial of 100 genotypes, you are evaluating in 10 locations, and then you are trying to select the top 5%, right? So it looks very simple from that perspective, but then if you actually do it practically, then you experience that in some locations you didn't have the seed. So the data is not there from that location. So two means, one mean which is based on 10 data points, another mean which is based on 6 or 7 data points are not comparable as such. We cannot compare these two and make a selection. So then we will never get to the true genetic value understanding. So that's exactly why we are talking about BLUPs. BLUP stands for Best Linear Unbiased Predictor. So what it does is the mean that is calculated based on only 6 data points that gets adjusted. So it kind of shrinks. It will either increase or decrease the value of that individual as against another individual that was evaluated in all the 10 locations. So this is one program which is developed by CIMMYT, the International Wheat and Maize Improvement Center. It's available freely. It's available in public domain. So what Ramu is going to demonstrate is how you can do this in a practical context. So many of you may be working on QTL mapping or genome-wide association studies or even line by tester study or diallele study. In all these cases, you need to use BLUPs if you are doing multi-location evaluation trial. So I think it's good to learn this one. So one is adjusting for that imbalance in number. The another one is the spatial variation in your trial. So in a big plot, in one corner you have certain plots, in another corner you have certain other plots. What is the heterogeneity, soil heterogeneity, which is causing conducting the experiment? After conducting the experiment, if you go to statistician, he cannot help much. Okay, so this one is one program that you can go back and practice also, because there is a practical data set that you can actually use in your homework.
And also the BLUPs corrects the each treatments or location may be having the errors also that can also be taken care to calculate the BLUPs. So here the meta look like this. So there is only the two buttons here, open file and the help. So open file is to help open a file of your phenotyping data, and help is if you want to read manual or anything is you can do available here. So I'm going, because of the time, I just go directly to, I'm opening a file. This is a comma separated file. This is Excel and saved as a CSV file. So you can open directly here. And these are all the 5 or 6 are phenotyping data: grain yield, silk, maybe plant height, ear height. I don't know the other one. So if you may be having a lot of data. Error correction, it will do the spatial correction and any other errors also. It's basically, BLUP is basically estimating the random effects compared to the another concept called BLUs. BLUs affects the fixed effects. So in this example, when you run the code, when you run the software, it gives the both BLUs as well as the BLUPs. So if you go to the top, so here is the experimental design. So as Babu highlighted, before you need to do the experiment, you should consult your statistician whether which experimental design you are doing, whether it is the RCBD or it may be a lattice design. You should know before you are going for planting. So for example, here it's a lattice design, for example, and if you're covariate example, say for example, the plant height is highly correlated with highly affected with plant yield. Right, so you can add, if you have the covariate, you can use that covariate, and here the genotyping factor. Say for example, I'm doing by management, means it's a individual location. It's not individual location, it's a practice, whether it is a drought or control, or it is a low end or a high end, whatever may be this one. And environment is basically site, that means where your location is there. And the replicate, this is how many replicates you have in your experiment. So I'm taking this rep, and the block is a blocks information, and the genotype. Genotype is basically the entry. Entry means it's the genotype here. So once you select this one, then you simply continue here. So either you can go back or you can continue, just continue. It says that there is a output folder analysis one, it's already written. It says that it's already existing. If you want to save your data output, you can write the different name here. So here, if you say 10, for example, so then continue, then it's move on to the next page. Then Analysis options. There are plenty of analysis options like correlation, environment, and correlating variables, and you can calculate the BLUPs and BLUs. Today I'm going to show you only the calculating the BLUs and BLUPs because of the time, and select this BLUs and BLUPs. So I want the BLUs and BLUPs at individual level, or you want at management level or environment level, but I would like to have the individual level. And here I would like to select which traits you want. Say I would like to select yield, cell king, font height, here height, and whatever. So you can select all this one. Then select all, then drop, or whatever you want. If you select and drop, you can take off. Then analyze the data. So it's automatically analyze the data. When it's analyzing data, you can see there is a bar is going on. It won't take much time to calculate the BLUPs. So behind this is already all the R code is running behind this. Okay. So all these variables and all this data is put it into the code and run the data. So analysis successful. I said okay. So here you see on the left bottom panel here you can see what is the R code used to run calculating the BLUPs and BLUs here. So here there is a big code here, and it also gives a lot of other information like here. So in environment, say in the drought condition, so these are the trait and these are the genetic variants and the heritability in that particular trait in that location and what's the p-value. Like that, you will get information for each and every environment or every treatment or management practices here. So you will get like this. Then you can, if you want to see how this look like, just simply open the data. So it will open the data. So from all this, you have the reps, you have the site location, say 10, and each location has 2 reps. You have the 20 data points for each individual. So all this will be taken care to have only one value for each individual. You see, in this drought 10 management practice, genotype 1, the BLUP for green yield is here, and the blue for green yield is here. So the BLUPs and BLUs, where is mostly the difference in calculation is how your model or the variable is fixed, whether it is a fixed or random, or whether you are taken into the consideration of the genetic variance presence inside the material. So that's how the difference between the BLUP and BLUs. BLUPs and BLUs are mostly highly correlated. If you take any one of them, you can take most of the things, but mostly we rely on the BLUPs. So once you calculate these BLUPs, you can just copy. So these two as a phenotyping and genotyping data for your genomic selection purpose. So is this clear or any questions? Yeah.
So BLUPs and BLUs are correlated as long as your data is balanced.
Yes.
So if you have 10 locations data on all the 100 genotypes that you evaluated, then no problem. BLUs and BLUPs, if you rank the genotypes based on BLUPs, or if you rank the genotypes based on BLUs, the ranking will not change if the data is balanced. If the data is imbalanced, that means some entries are evaluated in all locations and some entries are evaluated in incomplete number of locations, not 10 but 6 or 7. If that is the case, then you might find a difference in the ranking. Okay, so because the shrinkage happens on 2 accounts. One, the balanced nature of the experiment. Number 2, your spatial analysis.
Yeah, so that's how you calculate the BLUPs and BLUs. So here you can directly take the values to put or feed into the genomic selection model. So I calculated and I copied directly from here and put into the phenotyping data for that particular traits to run the code for the genomic selection. Is there any question? Because I'm done with the BLUP. So is there any questions? Huh? BLUs? Yeah, it's already there. It's already taken care because it's based on the management. It means drought or yeah, only one is there. Yeah.
Yes.
Yeah.
sir, does it account only for the experimental designs, RBD and lattice?
Yes.
Does it account for any other experimental designs?
Yeah, so far it's only for two designs. If you need, probably you can write back to the Meta people. There is an email available for this one. If they can upload, maybe done. Yeah. So far it's supporting the lattice and RBDs.
Okay, so this is for only one environment you are doing. So if you have multiple environments?
Yeah, so basically site is multiple environments. Okay. Location.
This is actually
Okay.
multi-location data.
Multi-location trial.
Yeah, but when we selected the variables, so I got a confusion there. So
No, so all these analysis actually make sense for our multi-location data.
Yeah,
so
yeah.
if it's a single one location data,
Right.
so even simple BLUPs will do. So BLUPs come into picture when you have multi-location data and you want to correct the analysis for the imbalanced nature as well as the spatial analysis.
Yeah, any question?
I think we can move to the next
I can
one.
move? Okay. So, yeah. So as already Bob mentioned, this is mostly useful for the unbalanced design. And I will move on to the next one. So next is genotyping data. If you to run the genomic selection, you need the genotyping data. So this is a format data look like. So first column is the SNP ID, and second column is the chromosome, and third is the position where it is, and the ABCD 1, 2, 3, 6 up to 6 is here. Basically, this is the individuals or the genotypes, and this is a 0 and 2, 1 and 2 are scored as alleles. Alleles are scored in the format of numerical format. So 0 means 0 either allele 1 or the parent 1, if you know the population, the parents, and 1 is equal to heterozygote, and 2 is equal to allele 2. So at a given point in a given SNP, there are only two alleles, either allele 1 or allele 2, or to have the two allele combined together in a heterozygous. So in that format, 0, 1, 2 will be made into this matrix, and this matrix will be fed as a genotyping data in the genomic selection model. And the genomic selection model, so I'm going to demonstrate on R programming. In R. So this is R is also publicly available and free. Anybody can download. So I request all the students to learn the R for statistical analysis. This is a very, very powerful. A lot of models are available here. So can you raise your hands? How many people are already using R or learning R? Not bad, but still I would like to encourage all the PhD students, at least they should learn one of these statistical analysis program, so they will be benefited for their career. So here in R, this R is, when you see the window, there are 4 different windows. The first topmost here is the code where you can write the code and save it. And here is the one where this is called console, where the code is run and all the results will be displayed here. And this is the one where your environment, your factors or the data will be stored. Variables will be stored here, and you can see what is that. And here, if you have any plots, and you can see the plots on the extreme right bottom corner. I would like to request all these right side people to come this side, because whatever I'm showing here will be available only extreme left bottom. Okay, I'm not sure whether you are able to see that. Are you able to see that on the extreme bottom, the cursor? Okay. Otherwise, you can. There are a lot of chairs here. You can move this side. So, foremost is you need to. So first foremost is that you need to know where your files are located. So that is called is a working directory. So you need to change your working directory. and you need to write the code where your files are there to read. So that when you run this line, that means your R analysis or everything doing, it's only saved in that particular folder. And before that, you need to know which packages are going to use in your analysis and whether that package is available or not. If not, you need to install. Here I commented this. The first character hash symbol there is basically the commenting the code. So I commented that because I already installed and loaded this package into my program in my computer. So when I run this one, so I'm changing my directory to that particular place in my computer where all this my data is available. And here, this is reading this marker genotyping data. So here it says marker full data as a matrix. I'm reading as a matrix and data file, and the genotype one, the header is true. As I show you that I have the header like SNP, chromosome, position, and ABCD 1, 1, 2, 3, something like that, right? So this is a header. That is not really the data. That is a header. I have the header. I would like to tell the program that I have a header. So when I run this one, it takes a little bit time because the genotyping matrix here is huge. It's having approximately 50,000 SNPs data, 50,000 SNPs and 200 individuals. So it's already read the file. Then when I after reading the file, I need to make sure that whether the data is read in the good right format or not. So I would like to execute the command called dimension of my variable. So it says that 49149 and 284. That means it's a two-dimensional data, and there are 49,000 rows and 280 columns. So when it's read, I need to know that yeah, this is correct or not. So I know that this is correct. This is my number of columns and the number of rows in the data. So okay, it's read correctly into the program. Then the phenotyping file. Again, phenotyping has a header, so I put header is true, and I need to make sure that header is read and the dimensions are correct. There are two columns. One is a sample ID, another one is a phenotyping data. So here I'm doing the phenotype is called color, where is each individual, whether do you have a color or not, is scored as the 0 or 1. And here there are a couple of codes here I will run through because in the data analysis, in the SNP data, you have the SNP chromosome position. Those are not really the genotyping data. It has to be excluded. And in case of the phenotyping data, all genotypes may not have the phenotypes also sometimes. So I need to correct this. All these individuals existing in the genotyping data should match with the phenotyping data or vice versa. So I will run this code to make sure that it captures only the data part. So here it's calling the number of columns, and it captures the IDs for each column, and it combines the IDs and the column names, and the markers matrix, and the phenotyping data, it's taking only 1 and 2 columns, and the number of markers and the number of phenotypes. Now I'm going to run the one for loop here where it checks whether given individual in the phenotyping data file exists in the genotyping data file or not. Once it is executed, then the dimension of your data set will be a little bit changed. Once it is changed, whether you need to make sure that it's changed correct or not. So the code is running. Okay, it's finished. Then here is the dimension of my phenotypes, and taking the phenotypes again, and extracting the markers information, and make sure that whether the markers information, I have the 278 individuals. Here previously I see the 284 individuals, but now it is 278 individuals because there are 6 individuals where you do not have the phenotyping data. If those individuals are taken off in both cases, then it is available like this. Then converting the markers data into a data frame and the phenotyping data into the data frame and converts that as a matrix. And here this particular line of code, it's taking the transpose of the matrix. That means you have the rows as a SNP data, SNP IDs, and the columns are the individuals. But in your phenotyping data, you have the rows, the genotypes individual. So I need to make sure that in both data sets, rows are the genotypes. So to do that one, I'm transposing the genotyping matrix. So it makes the rows into columns and columns into rows. And I'm taking another variable called y. Y is yield in terms of yield, but otherwise in any phenotype also you can call this as a y. And x is calculated. X is taken as a transpose of the matrix. And now I'm going to run the Bayesian regression model. All these Bayesian models requires some prior information. So I'm going to run the code, how to calculate the priors. So this is the code to run the call the priors, and I'm going to use the number of iterations is 300, and the burnings is basically the bootstrapping. Here I'm doing this 100. 100 means 100 individuals will be taken out and it will be done. Okay. It's time up. Okay. So I loaded the package and I loaded the PGRR package. That's where you have the model, and phenotypes and genotypes is loaded. And this is my FitBR is the model where I'm running the code. You can see that a lot of code is running and it's moving up. So that means it is running the code and it's developing the training population, and it's finished. So when it is finished, I'm trying to capture the genotypic SNP effects. Then you can see the SNP effects are calculated and populated here. So if I want to zoom here, these are the SNP effect. You can see there are a couple of SNPs are coming on the top. That means those particular SNPs are having much, explaining a lot of variation in the data set for this particular trait. And if I want to see how much the phenotypic estimated phenotyping value with observed phenotyping value, then you can see the correlation with observed versus expected. That is around 97%. That means the model did a good job. So here I didn't do any, I didn't separated the data into training population and test population. So all the data will be seen in the model. That's why you have the very high accuracy. But when you run actual model in 10 times, in 10-fold, you can download this GEVs as well.
Ramu, what was the phenotype?
Color.
Color. So the phenotype used is color, which is very simple in genetics.
Simple traits. That's why you have the very high accuracy.
That's why you are finding a correlation of 0.97. So that means you are able to predict very well. So if there are 250.
So 10-fold cross-validation means, say, for example, I have the data and I divide the data into 10 different segments. So in each column is each fold. So in each fold, whenever it's running the fold, that particular segment, one of the segment will become a test, and the remaining 9 will become the training. So that means we are making sure that at least in the model, at least each genotype will fall once in a test population. So that's how the 10-fold cross-validation means.
So maybe in the interest of time, Ramu, I think we have another presentation, and then Vice Chancellor's session will be there valedictory. We can't postpone that session. Can you show the graph?
I will show you an example. I already run it.
Yeah.
So here, this is another example here. Here, this is two traits, the flowering trait and the yield actually. So these are mostly, both are controlled by the many genes. But in case of the flowering time, you have the less number of genes controlling compared to the yield. That's why you are having a very less prediction accuracy for yield compared to the less complex trait.
Now, do you have that graph of linear, the correlation wherein it shows the predicted versus observed?
Yeah, it takes time.
Takes time, okay. So you have the results already? Or you don't have?
I didn't say.
Okay. That's fine. So okay, anyway, I think this session is not that in this half an hour you'll be able to do everything, but it is just a primer so that you can go back and start exploring whatever publicly available information is there out in the domain, right? So what we will do is we can actually provide the data to maybe the coordinator, and then he can circulate it to whoever wants it. You can get the scripts as well as the genotype data as well as phenotype data. Everything is available for you to explore. Just play with it. I think over time, you will learn about it. Thank you very much, Ramu. Thanks.